So let's go. So I'm Grégoire, and with Renaud, I've prepared this presentation for AI4Belgium and the specific AL's webinar that Roald and AI4Blackbelt has organized. On the question is your AI useful if nobody can use it, and how to effectively bring your AI in the field of expert life, and with examples in the field of digital pathology. So who we are. I'm no more CEO, but CMBDO at Cytomine, so Chief Marketing Business Development Officer. And Renaud, which helps me to prepare this presentation, is the CTO of the company. And Cytomine focus on collaborative biomedical imaging. And before starting on more what is AI applied to this discipline, I will focus on what is biomedical, because Frederick just went to speak about medical imaging. So biomedical, what is this? It's more focused on biomedical samples, for example, liquid like blood, where you're gonna observe and take images from the blood, from the cells and all the different components to sort them, to classify them, and to count them, for example. But also cells, when you are doing cell culture, it can be human or animal cell culture, but also fungi or bacteria. And the same game is there. You have to identify all the cells and count and everything. But the most of our job is to focus on biopsies. So a biopsy, it's a small pieces of tissue that has been taken off a patient for a confirmation of diagnosis when there is a doubt about, for example, a tumor in the cancer, to see its aggressivity or if it's benign or malignant and so on. So this piece of tissue is processed by laboratory technicians to prepare it to be seen by a pathologist, and you're gonna slice this sample in very thin layers from a few microns. Then the pathologist gonna see this very small slice under microscopes to see what are the cells, how they organize, and so on. But if you don't prepare correctly your sample, you will not see everything. The image will be pure white and you will see nothing. So for digital pathology compared to medical imaging, you need a lot of preparation of the samples. And first, if you have to stain it. So here it's a piece of the skin of your finger, for example. So all the wavelets here are your fingerprints. So in red, you have your skin, and all the green and pink part are the tissue under your skin. So it has been stained with Macron's trichrome for put some structure in evidence. And if you change the coloration, if it's a PES, periodic assay shift, you'll see some other components of the cells of the tissue and so on. So the samples must be prepared. And if in radiology and medical imaging, classical medical imaging, you are working with a shading of gray, it's in digital pathology is definitely a full colored world. So the big difference also with the radiology is that the pathology is a very lonely work. Why? Because when you observe some sample under a microscope, the slide is very thin and fragile, and it has been very difficult to obtain the sample. So you don't want your slide to get broken. So when you need a second advice, for example, to confirm a diagnostic, but also in research, when you want to better understand what is occurring in this tissue, it's very difficult to share the information, to share the slides. So generally, it's a pathologist which move to close to his colleagues and which use for quite funny adaptations of microscopes with like this dual head here, which is able to see the image, but not really to collaborate and add some useful information. So from 10 years ago, it was because digitalization of pathology is very recent compared to medical imaging. So 10 years ago, we have seen the first robotic microscopes and then the full automatic scanners to transform these samples into digital images. And the hope was very big to be able to share them and to work in at the same times of the same sample and to enhance collaboration. But here is an example. These are two lungs of a mice. If you look at your fingers and you look at your nails, it's quite the same size, so 15 by 15 millimeter. But when you scan it with a scanner which has the same resolution of a microscope, which is actually 200 nanometers for 1 pixel, it will give you a very huge image. And if I open this image on the classical resolution of my screen, it's like if I have to explore 2 tennis courts with my screen. So the game here is not only the complexity of the image, but also its size. And if I have to compare the role of pathologist and a radiologist, it's like if a radiologist, you ask him to open 256 thorax X-rays of human at the same time in terms of surface of image to analyze this. So the challenge here is very big, and it has an impact also on the weight of the images. The images are between 100 megabytes to sometimes more than 100 gigabyte only for one image. So such big image, you cannot share them over internet as easily as an X-ray, MRI, or a picture of your holidays. And you don't sometimes even are not able to open them on classical computers. So the pathologist needs to have a way to make them travel, generally on a hard disk, and have some supercomputer to make them open. So the collaboration is very difficult, and somehow it's drive to no collaboration anymore. And if you succeed to do it, you have to invite your colleague, and no more too close to the microscope, but close to the supercomputer. And it's the same when AI enters the game, because these images are very complicated to get connected to with some AI libraries, and it's not possible to work in remote. You also have to be on the same plane. So at Cytomine, we are focused on energy since 2010 at University of Liège to build a collaborative biomedical imaging solution. And the idea was to move all this technical pain from computers of pathologists to web servers. And on servers, it's very much easy to get skilled and to have sufficient power and storage to play the game. So we provide a web interface like this one, where you can open this image and zoom inside and work like if you were in a microscope and add some information like annotation and so on. And like this, people can work on these images like you are sharing, for example, big image with your mass of Google. They can work at the same time, anywhere and anytime, and in one-to-one, but also in team. And we have succeeded to make it in a MOOC, so a massive open online course for up to 4,000 users at the same time on the same sample. So it proves that it can be used by a lot of people at the same time, as it's also used at university for teaching for the same reason. And the game, it's also facilitated for AI engineers. So the game, it's also quite easily to get have only one point of connection for the AI to develop, and you can develop it on your side and push your model on the servers when you can have all the power and storage necessary to make them run and so on. And you can also work with a lot of colleagues using some features that I will present. So concerning the AI applied to digital pathology, for most people, it's just still a black box. So the game here, it's I have a good news for you. AI is no more a black box. AI is in fact two black boxes. The first one is the training phase. So in the training phase, you take your data, you take the metadata, or what is called the answer here associated to the data. So what are the classification and the object inside and so on, and you make them in contact with a model architecture. And the training phase is starting to build a model, and this model, this black box, should be considered as a gray box, because you still have some control on it. You choose what data you put inside, you choose what is the labels and the information, and you can choose what are the model architecture. But at the end, when the model is trained, you have to use it as this, and you just put new data inside, and it will give you some answers regarding to your questions. But what occurs inside still remains a black box. And to understand how to have a good AI, you can use this simplify drawings. And the principle is well known. Is if you put garbage in, you will have garbage out. And if you put diamonds in, you will have diamonds out, but only if there is diamonds everywhere. So you need to have good learning algorithms. You need to have good training data, and you will see what is good training data to have a good model. And after that, you have to use these good models on goodly prepared new data, which are consistent to the one which has been used for training, to have a good results. So in terms of models, there is a lot of model actually that can fit these big images and the way they are built. So all the AI engineers now are very well knowing how are the different models that have to use regarding of the questions that is asked by the scientist. And the game, it's very much more complicated, just sort of cats and dogs, as Frederick has explained before. And because the images that we have here, it's very wide, as I explained. So here is just a very small part of this image. And in this image, you have to compare objects, which are cells here, which are organized in tissues. And the difference between a normal cell and a 6 cell will be very, very thin. And sometimes there is no difference at all, and it's just a question of how it's organized in space and so on. So you have to pay attention that your AI will very fit the tissue and the disease that you wanted to see. Regarding also of the coloration, because some tissues can be very different one of each other, regarding of spatial structure and coloration and so on. So here, for example, the game gets simplified by use of a coloration, which color in brown all the nucleus of the cells where a specific gene is expressed, for example. And you also need to have a lot of data. A lot of data need a lot of high quality images, but also a lot of annotations to train this AI to be able to recognize what you want it to recognize afterward. So it can be segmentation of zone like here, when there is some inflammatory process may occur, but also tagging of cells and so on. And you must avoid also all default in the sample preparation. Here, the slice of the sample has been sloped, and this can be driven to an incorrectly trained AI, but also as the samples will be acquired with the scanners, you can have blur, you can have so, and the coloration robot can be out of date, so the coloration might not be consistent and always the same and so on. So there is a lot of default in the image that can drive AI to be incorrectly trained. So there is a quality process that must be put inside. And when it's going to development of AI, you just may also pay attention to the staining, because regarding a same pathology with the same type of sample and so on, if the slides have not been stained in the correct manner, the AI will not be trained on correct data and will not be able to use on the same kind of tissue for the same pathology in another hospital, where they choose another staining to make the diagnosis. So there is also a wall between the AI developers and the images, because as it's sensitive data, it's generally locked inside the hospitals where they are being produced, or inside of the laboratory when it's data which more relies on research and so on. So we also need to have some bridges between the AI developers and the pathologists to be able to develop this AI. So how to effectively bring AI in the field expert life? First of all, it's an observation, it's an advice that we say to all our customers. It's don't let only the AI developers responsible of the training phase and only the end users just use AI at the end. You must take the end users in the training phase also, and to manage with them all the quality process of the data, the quality process of the metadata they add to the images, for example, and evenly to show them all the different intermediate results during the training, to be sure that at the end the trained model will fit exactly what the pathologist needed. And mainly because it's not only one algorithm, generally it's a collection of one which calculated some different metrics that it's needed for a diagnostic, which is very widely and more global afterwards. And for the wall between the both, we need some regulation, but we need also some technical tools to allow the sharing and the connection between the AI scientist and the pathologist to enhance this real collaboration between these two walls. So it's also needed a large collection, image and annotation, as I say, but it's also a question of security to be sure that no data is leaked anywhere, but we need data to train the algorithm. So we must develop this kind of tools. And for that at Cytomine, we also pay attention of having an API to build a technical bridge between the two worlds, the world of the pathologist with the screen and the world of the data scientist with the access to the storage and the computer centers. That's why we use API built in Java, Python, and open standards and open formats and so on, to be sure that we can get interconnected with the data of the scientist. And it's only at these conditions that you will have this cooperation between these two levels of expertise. And you also have to pay attention on the user-friendly aspects. I had these questions pop up in the previous sessions. This is what is considered as a user-friendly front end for a developer of AI, and you may notice that it will not fit the need of a pathologist. So when you have to run an AI, it's only, do not let the job only to the AI developers. Please provide user-friendly interface also to the pathologist, able to make the run by themselves and to see the different results by, for example, changing some parameters at the end. And you also have for sure to have user-friendly interface for the end users. In our job, it's pathology. So here you have the same images for the region of interest on the left, where the AI has been run, because we are also the possibility to run the AI only on a short part of the image to not consume too much energy. And in green on the right side, you have all the nucleus of the cells that have been identified by the AI, for example, and based on that, it can calculate metrics like numbers of cells and numbers of cells by surface, numbers of cells in mitosis, and so on, to give some information to the pathologist to be able to bring his diagnosis. But the diagnosis still is the responsibility of the pathologist, and you also have to give them some tools to sort the results, to be sure that they really understand and give all the metrics that they need to build the diagnostic. And these are the only conditions to make a full diamond chain with a good data, with a good model and a good result, because everyone in the chain will adopt the solutions. Both the pathologists, which will be happy because the AI will bring real answer to the scientific questions and help them to make better and quicker diagnosis, but also both AI engineers to help them to provide this good AI to the end users too. So if you have more questions about how to use AI in digital pathology, we are Cytomine, and please get in contact or ask your questions in the Q&A section. Thank you very much.